in chapter 18, where we're talking about the blood and the formed elements of the blood, and we had worked our way up to talking about the production of what we term some of these formed elements, which at this point are going to be those red blood cells, the white blood cells, and the platelets. So we left off at this point, and we mentioned that there is a huge stem cell that will give rise to colonies that will give rise to the remaining cells, including those that are the red blood cells, the white blood cells, and the platelets. So at this point, for the red blood cells, we were trying to point out on Tuesday that their important function, carry oxygen, even though it can, and they will, pick up some of the carbon dioxide, their major job is to carry the oxygen. Because if we deny any of the tissues, which means at the cellular level, if we deny the oxygen, those cells are going to begin to die quickly, which could lead to death of that tissue. So one of the things that the red blood cell will contain, they're going to contain this enzyme so that that carbon dioxide, rather than it having to be picked up in the red blood cell, it can help buffer the CO2 so that we don't create a pH problem in the blood, okay, and therefore transport the CO2 to the lungs so that we can breathe it out without affecting the pH of the blood too much. So the red blood cells, even though they're going to have the ability to do that, their major job, transport the oxygen. The little bit of CO2 that they're going to pick up, in comparison to what's going to end up getting buffered in the blood, that's going to be more in amount than what the red blood cells are carrying. Because red blood cells, when they're carrying the oxygen, they may be carrying 100% of oxygen molecules, but when they get to that tissue level and the cells, they don't have to drop off all of their oxygen. So to give rise to the red blood cells, we're going to see that we start with the HSC, which is that hematopoietic stem cell. That stem cell will give rise to a colony. That would be the CFU. I know that doesn't sound right, but the colony forming unit, okay? So from that huge stem cell, which does give rise to all of those red blood cells, all of those white blood cells, the platelets, okay? For us to actually get the red blood cell, which is the erythrocyte, that stem cell gives us a colony. This is going to be a colony-forming unit. This one gives rise to an erythroblast. What do you remember about a blast?
Builds up something.
It is to give rise to something, to help make something, okay? So that this is our immature form. The goal is, with a red blood cell, produce this blast. Now remember, this is taking place in the bone marrow. Give rise to the blast, which is the immature cell. It will get into the bloodstream. It will spend roughly 2 to 3, 3 to 5 days, not very much, okay? Kind of just moving around in the bloodstream in the immature form. So roughly about the first 5 days of the life cycle of this cell, for about 5 days, it's going to simply move around. It will still have its nucleus. It will still have its organelles, because its goal, produce hemoglobin, all right? And we talked about that particular gene on the DNA, okay? So, produce the hemoglobin. Second, make that enzyme. After roughly about 3 to 5 days, spit that organelle out, spit the other organelles out, so that the only thing it's doing now, carrying that material. And then it becomes our erythrocyte. It becomes that red blood cell that is now going to transport oxygen, which would be a mature cell. At that point, it has roughly 120 days to do what it's going to do. And it takes about 1 minute for a red blood cell to make a full circuit around the body, from the time it leaves the heart until the time it returns. The hemoglobin that gets produced. Do you guys remember us talking about when we begin to have that process of making a protein? Remember, in the process of making a protein, where you're going to have transcription, you're going to have translation, okay? As that is occurring and the translation is taking place, the first thing that happens is amino acids get strung together. Does everybody remember that? Did we talk about that? Okay, so in producing that string of amino acids, don't forget they're held together by peptide bonds, okay? Now, once you have strung together all of those amino acids, do you remember what structure of a protein that was? Our primary structure. The primary structure is simply the stringing together of the amino acids. As that structure continues, those amino acids can fold or bend. Do you remember what that structure was called?
Secondary.
Secondary. If the secondary folds in on itself, what was that structure?
Tertiary.
Tertiary. Hemoglobin. When you look at hemoglobin, it is 4 tertiaries together. Okay? So they're trying to represent it here by our alpha and our beta chains. So we have 1, 2, 3, 4 for this structure. Now, structure determines what for a protein?
Which protein it is?
Structure determines function. Is there anything that can go wrong with hemoglobin?
Something like sickle cell.
The sickle cell anemia. Now, I want you to note the chain. All right, now, and let's just think about this for a second. We're going to have our DNA strand, and y'all know it's double-stranded. I'm only representing 1/2, okay? Located along that are our genes. It is the gene that codes for a protein. That's the only thing it can code for, okay? When it talks about 141 amino acids long, okay, that means that the gene for my alpha chain, okay, is only 141 bases long out of a chromosome that is millions of bases long. But the only thing that the red blood cell needs to focus on is that gene. And then my beta chain, which is 146, and then my gene for the carbonic anhydrase. Is everybody following me? Okay, so during that 3 to 5 day lifespan, what it's doing is it's continuously making this. All right? At the end of that 3 to 5 day time frame, the organelles are spit out and it can't make any more. Are you following me? Now, the reason I'm pointing this out, sickle cell. Sickle cell does what to hemoglobin? Does anybody know? Changes the shape. So, if it is dependent, if the shape of the protein is dependent upon this coding, this is the point it goes wrong. On the beta chain, the 6th base, at the 6th base, it's wrong. You remember the bases, A's, T's, G's, and C's? Okay? That base is wrong, which means that it codes now for a wrong amino acid, which means that as they're strung together, the folding now changes. Does that make sense? That one base being wrong leads to a debilitating disorder that can range from being mild to severe, and it affects multi-organs. That's how important it is that things are coded for correctly. And that's it. That's all that red blood cell is focused on doing. The goal is to create it's going to create way more, but to get a quaternary structure. Do you guys remember quaternary? Where it's 2 or more tertiaries together.